Good morning, everyone. My name
Morning.
is Dr. Yeo. I am the director of this summer course. And let's just hang on for a minute or two before we get started, just to make sure that as many people can come in as possible. I will be going through the introduction, and I'm going to put on my slide in a minute. Can everybody see my slide?
Yep.
Yeah.
Okay. Very good. Okay, let's get going. So as you can see, this is the summer 2021 lab management course. This is part one of the course. Some of you who came in last year as the first year is sort of doing this in reverse. You had the part two of the course. So now you're coming back to part one. But for new resident, welcome to pathology. This is probably one of your first courses that you're taking among the others that you have taken already in other parts of the lab. So there'll be 8 Wednesday weekly lectures. We try to do it as much as possible in a case-based study, but I cannot guarantee that everybody would do it. So the first 2 sessions will be taught by me. We talk about quality management, which is a really core principle for the clinical labs, as many of you will one day become lab directors, regardless of where your other position might be. If you are APCP resident, most likely you may subspecialize in one part of the AP, but you might also be asked to oversee some part of the clinical lab. So the quality management is an integral part of it. And then we'll follow by Dr. Van White will talk about utilization review and the send out tests. Renata is in our coding and reimbursement department. She'll talk about that. And then we have 2 people talking about financial aspect of the clinical lab. One of the very common feedback that we get from our fellow and our resident is that very little is taught about the financial aspect of a clinical lab, and a lot of times they'll go into a new position and all of a sudden they inherit a big chunk of trying to balance the finances and things like that, or at least be bombarded with a lot of financial terms that they're not very familiar with. So we have incorporated the financial aspect of the clinical lab into this series. And then Dr. Christensen will talk about the path informatics part of it. And then at the end of it, given that we have just started on a major migration to the Epic Beaker, which is lab information system software, that is a almost 2-year project that just started. I had 2 of the key people from Epic Beaker group to give an overview of the implementation. No doubt in your future career, you will also encounter aspect of a lab information system in terms of either implementation or changes, and it is good to at least have an idea what that involves. Okay, so that's just an overview of the whole course in this summer. So I'm going to proceed to talk about quality management of laboratory tests. I'm the director of clinical chemistry lab, one of the more larger lab in terms of the scope of testing, the quantity of testing, and the quantity of automation. So because of the fact that we can produce quite a number of tests per hour, it is very important to have lots and lots of quality safeguard in the lab so that when you run into issues, you catch it as soon as possible, because if you don't, you could produce hundreds of bad tests in an hour, and then it is a huge headache to try to come back later to try to correct the inaccurate result. By the way, if anybody have any question while I'm teaching, please don't wait till hold on. Did I get disconnected or no?
No, we still hear you.
Okay, I was suddenly like disappearing and no quiet. If you didn't hear me, please either text me, or let's say if I get cut off, please text me, and so that I don't just go on talking as if I'm talking and nobody knows what I'm talking about. Right. All right. Thank you very much. So let me make sure. I want to put a big screen, but let's try and see big screen. All right. Okay. So everybody can see my screen. So I put up a hypothetical case, which is actually not a real hypothetical case. It's sort of taken out of one of my experience a while back. So let's say you are the director of the clinical lab, and you were contacted by the vice president of your medical center community affairs to set up a health fair for the underserved community. So like, in a park or in some aspect, it could be, in a local community center or a park. So the vice president would like you to set up a free diabetes and lipid cholesterol screening test fair. So it sounds like a pretty straightforward request, but before you go pack up your truck or your van or whatnot and to get going, you might want to stop and think about what issues you need to consider. So here I just flesh out a couple of issues that you need to consider if you want to set up a free clinic or free fair for this. What are some of the regulatory issues? What are some of the personnel issues? Who is going to sort of you trying to recruit to do the testing, and what kind of testing platform or device would you want to use, and how would you go about getting this achieved? So these are some of the questions that I sort of pose out there. Anyone want to take a step? Are there other issues? Just maybe a couple minutes, one or two of you would want to add on.
Sure. I guess one of the first things I'd consider is, they say free diabetes and lipid cholesterol screening tests. I need to know what exactly kind of tests they have in mind. What degree of complexity are these? Just going to be wave tests, or are these going to fall under moderate high complexity? Want to know if there's any sorts of certificate or licensing in place already, whether or not you would need to apply for a certificate of registration, or if you're starting from the ground up, or if you already have some sort of licensure to operate under.
Mm-hmm. Yes, very, very, very good. Right to the point. So it's actually quite involved. It's not straightforward as you might at first blush think. So we would now go into some of the aspect of what constitute quality management, and then we'll come back to sort of round up this case. Yes. So obviously you want to be able to do this to make a contribution to the underserved community, but at the same time, you want to do it right. You want to do it accurately. You want to do it legally, right? The last thing you want to do is to do it and then later find out that you are in violation of all sorts of either the city rules, city of Chicago rules, or the IDPH rules, the public health, Illinois Public Health Department rules, and CLIA rules as well. So I'm already throwing out a lot of words that some of you may not be familiar with and may be hearing it for the first time. So, you definitely don't want to do it the Elizabeth Holmes way, all right? A lot of you know who she is. She, a few years ago, she came up with this so-called revolutionary lab that supposed to be so revolutionary that you can do all tests with a couple of drops of blood, and so therefore, you know, upend the world of laboratory testing. We're going to put all the big reference lab out of business, and maybe even hospital lab will be affected as well. So what they found out is later is that it was all lots of, it's basically fraudulent. She never had the testing platform that she claimed to be, and she was using all sorts of cloak and dagger type of operation, and they have to do massive recall of the test later. And if you want to know more about it, just go talk to Heather. I think she was in the middle of that, but no more. All right. So hopefully we will talk a little bit about the regulatory aspect, the test categories under CLIA, what's CLIA, and then what constitute a good total quality management program. Yeah, and then as a part of the good quality managed program, you have to have very good internal quality control program. We will not be talking about this in this particular series, but when you come into doing your clinical chemistry rotation, your 2 months, some of you will be heading this way in September. We will actually go more into detail into internal quality control, and then also talk a little bit about external quality assessment. So I have 2 sessions here. All that I'm going to talk about is already given to you in the PDF, so you know you can refer to that. So what is an in vitro diagnostic product, or an IVD? Okay, so all the things that we have in our lab, whatever device or system or even reagent, okay, is by definition an IVD if the intended use is for health assessment, disease diagnosis, in order to cure, mitigate, treat, or prevent disease. Okay, so anything in the system, okay, it doesn't have to be an instrument per se that you can see. You know, when we talk about device, you know, we would like to see a portable device, or like an iPhone or something like that. It's a device for communication. But it could be just reagent, okay, that you buy, that you use to stain a slide, and then you read the slide, and then you make a diagnosis. If that's the case, then the reagent is considered an IVD, all right? And it could also be anything used in collection, preparation, examination, or specimen taken from the human body. So for example, when we draw blood into different kind of blood tubes, that tube of blood, that particular collecting device, that's an IVD, because even though it's not used directly to test the specimen, it is a conveying device that you are transporting the blood to be tested. So therefore, those tubes need to be regulated and needs to be cleared or approved by the FDA. So it's covered under FDA, is subjected to lots and lots of pre-market and post-market controls by the FDA. And on top of being regulated by the FDA, is subject to the Clinical Lab Improvement Amendment, or CLIA 88, that was first introduced in 1988. If you want to read more, just click on the link downstairs, or you could just Google search it. It's very easy. So that's an IVD. Any question here? All right. Don't just think that it has to be a physical box. Okay, it can be any component involved in generating the test result. What's clear? So Congress passed the CLIA in 1988. It's basically passed to establish quality standards for all lab testing. And basically the whole idea is to ensure accuracy, reliability, and timeliness of patient tests, regardless where the test is performed. So that's what CLIA is. And then it also defines what a lab is, okay? So a lab is any facility that does lab testing on specimen derived from human, not animal, not cell culture, okay? It gotta be from human, to give information for diagnosis, prevention, treatment, disease, and impairment assessment, blah, blah. So that's a lab. So this slide just gives you a little bit to differentiate the roles of CMS and FDA. Both are federal entities, and sometimes they sort of, you would think like they don't work together. Sometimes they appear to work against each other, but they're both trying to do the right thing, but they have their own charges. So in terms of CLIA, it is a usual fee funded. So it is the way that CLIA is able to run, although it's a federal entity, it collects its fee from all the CLIA certification it gives out to labs around the country. All right. So basically, from the fee, it used to administer the whole program. And CLIA program is really under the jurisdiction of CMS, which is the Center of Medicare and Medicaid Service, which assume primary responsibility for the financial management of the CLIA program. So CMS also regulate payment, for Medicare and Medicaid. So it's a really powerful body, right? So let's say if your lab fail to follow CLIA regulation, CMS could pull that reimbursement to your hospital or to your clinic or whatnot, and nobody can survive that, because still, despite of the fact that hospitals are paid by different, some of them, negotiate with different insurance company, private insurance, still a big payer is still CMS. For sure, University of Chicago being where we are, we have a big population of Medicare and Medicaid patients. We could not survive if we do not get reimbursed by CMS. One thing about lab tests is that whether it's AP, CP, kind of laboratory tests, they are sort of categorized in terms of how complex the test is, and they are graded into, moderate complexity, high complexity, and then they have the so-called low complexity tests, which are usually waived tests. These categorization is made by FDA. That's the jurisdiction of FDA. Because FDA, or the Center of Device and Radiological Health, regulates in vitro diagnostic, not just the laboratory tests in pathology, also in radiology as well. Okay, so they work hand in hand together. So, and in terms of CMS, it regulates all lab testing performed in human in the US. So they have the clear tools for this regulation, and all laboratory performing testing on human must have a clear license. You must have. If anybody runs human lab testing without clear license, they are in a violation of federal rules, and they could actually be persecuted and sent to jail for that. Any question? Anybody? No? Oh, there's somebody asking a chat question.
Oh, no, I think that was Heather agreeing with her sentiments.
All right. Okay, as I just to expand a little bit on the FDA role, it approved IVDs and kit, and in the kit, it approved all package insert. So, you know, especially in the clinical lab, whenever you buy an FDA approved test, the first thing that comes out of the box are the package insert, and tells you how to calibrate, what control, and what the testing procedure will be, what the suggested reference range should be, and what are the performance specification. And even if you buy an FDA approved test out of box, it is your role as a lab director to make sure that you do validation studies to verify that the performance of the test as specified in the package insert is met before you launch it for your hospital. So we do this quite as a matter of fact, and some of your colleague or your classmate already done chemistry can tell you that. If you talk to Clarence, he does this all the time, spending his time in chemistry, doing verification and validation studies, most recently for the COVID antibody test. So FDA, as I said, has the role of categorization of tests into wave, moderate, and high. In 2003, remember, CLIA was first introduced in 1998. That's why it's called CLIA, 1988. Sorry, CLIA 88. And then you know, many years later, in 2003, FDA combined the high complexity and moderate complexity into one category, but really not, because when they did that, they actually came in with subcategory to differentiate between the wave test within the wave and the non-wave test. The non-wave test will be, you know, so-called higher complexity test. So I don't know why they do that, but they did that. But in the end, we all in the lab still think of wave test, moderate complexity, high complexity. So in terms of FDA classification of device, they look at it based on risk. Okay, so subcategorization of the device could be under class 1, class 2, class 3. So obviously the highest class is the one that has the greatest risk of harm if the test or the device fail to work as intended. So that's why they need to come out with class 1, 2, 3 to be able to differentiate between the risks. So obviously, class 1 is the lowest risk, but regardless of which class it is, what is necessary in the package insert is always, you hear about what we call intended use. The intended use is sort of what has been approved by the FDA for the particular test or the assay that we talk about, right? Now, let's say, the intended use for a particular test is to monitor control of diabetes, right? Let's say, a particular test is, I don't want to name it, or for monitoring a type of cancer. Well, once it is released into use by the clinical community, individual physician could use the same test for a different indication. All right, because the FDA, while it regulates intended use, it does not have the right to regulate physician practice. So, if let's say a particular physician found out that this test could be useful for another indication based on some upcoming clinical research that been published, and they wanted to use it, they could, right? They could, and that's called off-label. So like medication, right? If it's approved for a certain disease treatment, and if that's been found to be also effective against some other indication, physicians have the right to use it, right? But the lab had to publish that this test is intended for this particular purpose. So, you want to meet those general controls. It could be suitable for intended use, manufactured under a high QA program. You must make sure that there is adequate packaging, labeling, device listing, and it's manufactured under good manufacturing practice. That's the minimum for a manufacturer to be able to market it. So I'm not going to read through all these words here. You guys can clearly read, but basically, the higher the class device, it is of a greater risk. So let me throw out a question here, because I don't want to hear myself speak for the next 30 minutes. Anyone can tell me if you have a molecular device, a PCR, used for identifying a variant, what kind of class device that would be?
I would expect that to be class 3. It's the sort of guiding, it may be used to guide therapy.
Yeah, exactly. So by definition, all the molecular tests, especially coming out of next-gen sequencing, real-time PCR, stuff that you have to make yourself, they're all like class 3 device if it's FDA approved. All right. There's another whole category that I have not talked about, which I talked about, is called lab developed tests, and I'll talk about that. But if any test that's approved for a particular indication in molecular, usually they are class 3 device, right? If something is, you have to implant into the body to get, constant reading, that's typically also a class 3 device. Okay, most of our tests are not implantable, but we are moving towards an era where we might be seeing some of those implantable device, if not already there. Okay, good. So a little bit more acronyms. You might hear about, if you deal with FDA, you might hear about PMA and 510K. I mean, this is like foreign language to me when I first entered into the field of laboratory medicine. But without reading all this gobbledygook here, the PMA is basically a category of regulatory box that FDA use to try to decide whether when somebody submit to them for approval, a device for approval, whether it's going to be a PMA, PMA means pre-marketing approval, or versus a 510K, all right? So basically, if the device is high risk, it will be categorized under PMA. I mean, that is the highest level of review for evidence that it is safe and effective for intended use. So that's why it's called a PMA. And FDA requires at least 180 days to make a determination on whether the device is going to be approved or not. So if it's being reviewed under PMA and is finally approved, it's called FDA approved device. Okay. I want you all to differentiate between FDA approved versus FDA cleared. Sometimes that term sort of confused between FDA approved versus FDA cleared. So FDA approved is devices that have gone through the PMA process. So the PMA process always requires the submitting entity to do human clinical trial. They have to do human clinical trial. All right. So which means it's more expensive. It's more time consuming, right? Because if you need to register, you have to do clinical trial, you have to recruit human subjects and things like that. So it takes a longer time. So that's a PMA, the highest level of review. So with terms of 510K, don't worry about how the word comes out. You know, who knows? The federal, the government comes up with all these terms, right? So the 510K does not require human trial to prove efficacy and safety, not safety. Sorry, typo here. So let's say all the manufacturer needs to do is to demonstrate that this device is substantially equivalent to already legally marketed device. So in the sense of like a Me Too kind of thing. If let's say, somebody came out with another device to measure whatever test already been out there in the market, okay? Let's say glucose, all right? Another new device to measure glucose. They just have to show that that new device is substantially equivalent to what's already out there. You can choose what you call a predicate device that's already out there being used. Okay, so therefore 510K is an easier route. The bar for clearance is much lower. You just have to show that it's equivalent, okay, by all the usual tests that we do, that is equivalent, that it will produce about the same result or equivalent result. And you don't have to necessarily do human trial to prove efficacy and safety, which is a lot of money and lots of time. So that's why the manufacturer like to avoid the PMA as much as possible. So you would see that in 2015, for example, FDA approved 3,000 devices via the 510K versus 47 PMA devices. So now you know why. Other than expense, it's also time. All right. Anybody have any questions here? 510K, PMA. Oh, you learn two new terms. All right. All right. So let's talk a little bit on the test category that I sort of mentioned in passing. Again, wave test, moderate, high complexity. So let me show you the latest categorization where they just looking at non-wave tests. So non-wave tests obviously are all the tests that we do in our clinical lab. We also do some wave tests as well, especially in the point of care lab. point of care environment. So again, even though they are non-wave, they still come out with a sort of a scoring number where they give 1 the lowest level of complexity and 3 the highest level. And so they just basically, total up the score. So if your score is around 12 and above, you are high complexity. And then if you are intermediate, you're sort of like moderate complexity. So in a sense, it's sort of like the old categorization. So it's like, as far as I'm concerned, it is high, moderate, and wave tests. All right. Now, wave tests are typically cleared by FDA for home use or outside of the lab use. Okay. The criteria is that the methods are so simple and accurate as to render the likelihood of wrong result negligible, or it poses no reasonable risk or harm to the patient if the test is performed incorrectly. So that's by definition, the FDA say, if your test can meet those criteria, we can consider it to be waived. And more of the new company, especially during this 2 years of pandemic, which is still not over yet, it's getting better. There is a push towards the lots of company, especially startup company, wanting to come up with technology that can be waived, so that they can actually sell it in places like the pharmacy. As you know, a lot of, as revealed by the pandemic, is like because people don't want to come to the hospital or clinic because they view it as a little bit more high risk, especially before the vaccination. Where do they get all their whatever needs they have? A lot of them goes to the pharmacies, the local pharmacies, and as shown by the vaccination, they also get the vaccination from the local pharmacy right now, actually. So there is a push towards revolutionized lab testing, especially the routine lab testing, to be done in a decentralized area in the pharmacy, local pharmacy. So that's why wave tests are sort of what the manufacturers are pushing to try to get into the hands of the consumer. Question there? Any question? Okay. Now, can anybody say that this is always correct? That even for wave tests, that the method is so simple that you cannot screw up? Anybody want to share? Have anybody bought one of these at their local CVS or Walgreens for whatever purpose? You don't have to reveal your purpose, but
No personal experience, but I mean, our clinicians have taught us that people will always find ways to do these incorrectly.
ways to do things incorrectly. I like that. Actually, some device that look simple, you not intentionally, if you don't follow the instruction, you could screw up the result. And I don't believe that if you go get a test, that if the test was performed incorrectly and gave you a wrong result, that it posed no reasonable risk. It depends on the test. And a lot of those wave tests do have potential, if not physical harm, it potentially could give you psychological or social harm. So as I said, there is no free lunch. So look at the test with wave method out there. I mean, there are a ton of them. I mean, if you look at it, chemistry, endocrinology, toxicology, immunology, I mean, can you say that if you did the HIV antibody test wrongly and you got a positive, when you got a false positive, that it's not going to cause some harm? I would argue that it could cause some harm, right? Depending on what the particular family situation is, right? So there's quite a number of tests with Waived method out there. Waived test means that you can buy it in the home, buy it for home testing, or it could be done by people in the pharmacy, untrained people, right? It could be somebody, because waived by definition, it can be done with somebody with just, maybe even a high school, just went through high school, they can do this test. And as long as they have manufacturer have given them some training, they can do the test. All right. And or you could buy home and do the test yourself, following the instructions. So that's the weight test. All right, the non-weight tests are all the tests we do in the lab, or the one that we're very familiar with. And we have just gone through a big CAP inspection, and that's what they look for when they come to inspect our lab. And the non-weight test category requires that the people doing the test have the knowledge, training, and experience. You have to hire a certain type of personnel. So one of the things that they look, the CAP inspector look for is personnel record, right? You cannot simply hire anybody from out of high school to do a very complex procedure. Okay, so if you hire somebody from high school, maybe the most they can do is to process a specimen, so on and so forth, but not the testing itself. And in California, in some state like California, the personnel that can do the test have to be licensed medical technologists, not anybody else. Okay, so they are highly regulated in California. So the medical technologies lobby is so powerful that they have changed the rules such that only medtech can run it. Whereas in Illinois, you could have medtech, you could have somebody with a bachelor's degree either in science or even in arts, but they major in a science, and you train them for a year, and they can do the test. So different states have different licensing or requirement for training and experience. The people running the lab needs to know how to make the reagents or prepare the material. You need to have SOP that characterize the operation step. You would make sure that there is a description of calibration, quality control, proficiency testing, and then you need to have a test system that can do troubleshooting and maintenance. Make sure that those are done. And in the end, your procedure should also make sure that there is some paragraph on how to interpret the result, and give them some sense of judgment when the results are out of control. So all this needs to be part of your standard operating procedure. So most of our automated tests in chemistry are of the moderate complexity type. The high complexity type are many manual methods, modified or lab developed methods, or methods requiring more interpretation. So all the stuff that you guys do in AP, where you go behind a microscope and look at the stain and make interpretation, those are by definition high complexity. There's no moderate complexity when you make slide interpretation. And then you write paragraph of what you see. So those are all high complexity tests. Most of our automated, as I said, in chemistry, and in some part of hematology, where they do it by like the CBC, automated CBC, diff, those are moderate. But as soon as they have to go into manual, that's high complexity. Right? Any questions? All right. Okay. All right. So now I'm going to talk about lab developed tests. So the lab developed test is a very hot potato area because it is an area of lots of controversy. But as university hospital labs, we typically are very, very protective of lab developed tests, because if we are not allowed to have lab developed tests, a big chunk of our tests will go away. Like a lot of molecular tests will go away because if we have to seek FDA approval, then we will often not be able to do it because we don't have the money nor the time, because at the same time we have to provide the lab testing. So that's the reason why we are very, very, especially the CAP is very view this as very important for our business. So the FDA defined LDT as IVD that is manufactured by and used within a single lab. Consider devices. So these are devices because it used to do diagnostic testing. So it has to be a device. And in the old days, we call it homebrew test. That term needs to has sort of being jettisoned because homebrew sounds like I don't know, sounds like Mickey Mouse, right? It's like you add a little bit of that, you add a little bit of this, and then you come up with a test. So the lab medicine community thought that's a bad term, so they call it LDT. So no more homebrew. So the CMS regulate and FDA do a very what I call it, a very sort of a dance around the LDT. So by definition, FDA has authority on all in vitro device. So LDT is an in vitro device. So by definition, FDA have regulatory oversight over LDTs. But because of the fact that no university hospital have the resource to get an LDT approved, the FDA sort of, even though it does have the authority, it sort of exercise discretion, meaning that it turned a blind eye on it right now. Okay, exercise discretion means exercise not to regulate it at this point. And the latest is, if you have been following the news, I think one of the senators is trying to get that authority to regulate LDT from FDA and give it to CMS. It's still brewing. So by definition, then, if FDA is not regulating it, then CMS regulate it to ensure accurate and reliable results. Okay, so the difference between LDT now, the way that it is regulated by following CMS rule, which is basically clear rule, is we just have to show that the test is analytically accurate and sort of meet the clinical indication as published. You don't really need to do clinical trial to be able to offer an LDT. If all of us have to do clinical trial, a lot of tests we could not offer, right? Because that's the difference. Remember, a lot of LDTs are high complexity tests. So high complexity tests usually is viewed by FDA as require require clinical trial for safety and efficacy. Who can do that? We cannot do that. We don't have the resource to do that. All right. So in terms of clear requirement for LDT, we just have to establish that it's analytical valid. All right. That means they need to be accurate, precise. You got to define how low can you measure whatever you are claiming to measure. You have to define that your test is measuring PSA and not cross-react with something that looks like PSA. So you want to make sure that it's specific. You have to define what is the reportable range. If it's a quantitative test, you have to provide some sort of a reference interval, meaning what normal people would run. So those are the basic for LDTs, okay? And it is limited to specific conditions, staff, equipment, patient population. And then you need to subject yourself to some sort of an inspection or survey. So CLIA usually use accredited bodies to do its inspection for them. Biennial means once in 2 years. So we use CAP. The difference between CLIA is that it does not include a review of clinical validation of the LDT. Remember, clinical validation means the FDA requires clinical validation before they clear a test or approve a test. And so that means that they have to do, clinical studies in human. So the fact that CLIA does not require that clinical validation makes it more palatable to the usually clinical labs in the university hospital or even reference lab like Mayo or ARUP or Quest or LabCorp. Even though they're much bigger, they have a lot of LDTs. They have a ton of LDTs because they can come in under these LDTs. Any questions here? Because I want it to be very clear to you guys what is the regulatory environment right now for the LDTs. Because chances are you as a future lab directors, you will be involved in making some LDTs if you're not already doing it right now. Anybody developing LDT in usually either in a lab that involve let's say you have a new set of antibodies that you need to develop to stain a certain tissue for a certain diagnosis of a cancer or some sort of a disease. You're making LDT. Any one of you? No? I would think some of you would have done that. Maybe you don't realize the LDT. Or if you work in a molecular lab where you are coming up with a molecular test for this variant or that variant, that's an LDT. Well, if not, you soon will be. All right. Here are the accrediting or licensing organization approved under CLIA. So these are agents of CLIA, right? Because CLIA, although CLIA itself can send their workers to come and inspect your lab, especially if your lab is violating all sorts of rules, like Theranos was violating all sorts of rules. CMS actually came in to inspect it. So they have a right to inspect the lab. So the typical one will be CAP, AABB. All blood bank needs to be inspected by AABB, even if we are clear CAP accredited. And then they have a JCO or Joint Commission of Accreditation. JCO usually inspect the rest of the hospital, and usually in our hospital or many hospitals that are kept accredited. When JCO come, they usually, if they know you're kept accredited, they don't come to the lab. They can, but they usually don't. And then for the there's another big organization called COLA, Commission of Office Lab Accreditation. These are the folks that usually inspect physician office lab, smaller lab in physician office. And then there are states where the state will come and inspect on behalf of CLIA. So they are called CLIA exempt state because like New York State or Washington State, they do their own inspection. So they are actually at a higher standard. So CLIA is sort of like if any state inspection has a higher standard, then they exceeded the CLIA expectation. So they are called CLIA exempt state. And some states have extra regulatory requirements like California, Florida, Wisconsin. And there are some also other local bodies that can do it. So we actually did 2014. Actually, we did a Chinatown Health Fair, and you look at all the people involved on the right hand side. These are all mostly Pritzker Medical School students that we got, and some of the lab people. So we actually have to come see all these devices here. We have to make sure that I got a CLIA license for this. So I sort of act like a CLIA holder for this, and make sure that the people running the tests are all lab techs here. These are all lab techs that do the testing. And the people giving the advice to what it means. We have a couple of physicians here, and I'll make sure that we have the CLIA license. I have to notify IDPH I'm doing. I have a CLIA license. But this is what we did. We did a health fair in 2014. There was kind of a lot of people. So I'm going to end here, and then we'll pick up the rest of the slide, talk about quality control and things next week. Any questions?
Yeah, I guess where are you going to start the recording in the PowerPoint slides?
Say it again.
Oh, so it looks like that someone is recording the lecture. Where is it going to be stored?
Oh, I'm recording the lecture.
Yeah.
So I usually record the lecture, and then I have, I don't know where I store it, but I will give you the link and you can pass it on to everybody.
Okay.
Okay.
That sounds good.
Yeah. Okay. No question?
Thank you.
Right. Thanks, Dr. Yeo. Bye.
Thank you. Well, you guys have a good day, and I'll see you all next Wednesday at 8 o'clock.
Yep.
All right.
Hope this has been helpful. Thank you. Bye.
All right.
Thank you.
Bye-bye.